video, we will discuss about the process of translation in case of prokaryotes. So what do you mean by translation? Translation is the synthesis of proteins from mRNA. In case of prokaryotes, the process of DNA replication, transcription as well as translation takes place in the cytoplasm. Whereas in case of eukaryotes, the process of DNA replication and transcription takes place in the nucleus, after which the mRNA is transported into the cytoplasm where the protein synthesis takes place. So let us see what are the components required for the process of translation. It requires the mRNA, which gives the code for the synthesis of proteins or for the synthesis of the amino acids. So AUG, which is present, it is responsible for giving the start codon. It codes for start codon. Next is the initiator tRNA. This initiator tRNA, this tRNA which is present, it helps in bringing the amino acid according to the sequence, and it is being recognized by the anticodon which is present in the tRNA. And for each code or for each amino acid, there is specific tRNA present. The third thing which is required is ribosomal subunit. Here we are having a 30S ribosomal subunit, as well as last subunit is present which is of 50S. And in case of prokaryotes, there are two types of subunits. The ribosome is of 70S type, and according to the molecular weight, it is of 50S ribosomal subunit and 30S ribosomal subunit. In the 50S ribosomal subunit, it is having rRNA as well as proteins. Here, it is having 5S rRNA and 23S rRNA in case of prokaryotes, and there are 34 proteins present. And in the 30S, it is having 16S rRNA and there are 21 proteins present. So the process of translation takes place in three steps, that is initiation, elongation and termination. Before that, we have to see what are the sequences present over here. In the mRNA, there are 5' end and a 3' end. So for protecting this end from degradation, the 5' end is having a cap and 3' end is having a poly A tail. And that is a ribosome binding site where the ribosomes comes and bind in this region. This is present in the mRNA, and in this mRNA, it is having a sequence from 5' end, that is AGG, AGG. Okay, so complementary to it is present in the 16S rRNA, which is A is UCC, UCC. Here 5' and this is 3'. Okay, this is having a complementary sequence in the 16S rRNA by which the ribosome can bind in this region. After 3 to 9 bases from the ribosome binding site, there is a start site which is present from where the synthesis of the protein takes place, and that is a stop site present here where the protein synthesis stops. Here there is protein 1 formed. Okay. Next is another one, RBS site, where the ribosome come and bind, and after scanning it forms from the start site, it forms a protein, and in the stop site it ends. Okay, in this way we can see that there is a 1, 2 and 3. There are three proteins which can form from a single mRNA. Okay, so we can conclude that from a single mRNA there can be multiple proteins formed. So let us see the process of translation. The first step, that is the initiation. For the initiation, it requires first the dissociation of this 70S ribosomes. The large subunit and the small subunits should be first dissociated. Then it requires the mRNA and initiated tRNA. So for this process to be done, it requires some initiation factors. They are initiation factor 1, initiation factor 1, initiation factor 1, initiation factor 2 and initiation factor 3. The first work of this initiation factor 3 is it comes and bind in the E site. So first of all, we should know what is the P site, E site and A site. The A site, which is known as, it is known as acceptor site. This is acceptor site. The P site is known as peptidal site and the E site is known as exit site. This IF3 comes and bind in the E site so that it will not allow the last subunit to come and bind with the subunit. The next work is IF1 and IF2. The IF1 will come and bind in the A site because it will not allow the first tRNA to come and bind in the A site. So now the P site is only left. So now the first tRNA will come and bind in the P site, and this will be bound by IF. The first is formulated methionine in case of prokaryotes. It is formulated methionine. It is not only methionine. Okay, why it is formulated methionine? Because in the F med, The peptide bond formation site is not present in the N-terminal end. Okay, so when the tRNA has come and bind here in the P side, it has formed the 30S initiation complex. After the formation of 30S initiation complex, the initiation factor 3 is released, and now the last subunit can come and bind in this region and form the 70S initiation complex. After the binding of this last subunit, the initiation factor 1 and initiation factor 2 also dissociate it and forms the 70S initiation complex. The next process is the elongation process, when the next tRNA will come and bind. Here we can see that the next tRNA comes and bind in the A site, and this has been done by another one in elongation factors. And after the binding, after coming of another one tRNA, it brings one amino acid, and this two amino acid forms the peptide bond. Okay, so here we can see that the EFTU which is present, it will come and bring the tRNA. It requires GTP, and this tRNA will come and bind in the A site. After the binding of this tRNA, after 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 the tRNA is in the A site, the AFTU is now released, and now it is in the GDP. Take any of the tRNA. Again, it will be converted into GTP form. So this work is being done by another one elongation factor, which is known as EFTS. This EFTU, when it is in the GDP form, it cannot work. So EFTS, it removes the GDP and gives the GTP. Now the EFTU, now it is active, so that it can take another one amino acid tRNA and bind in the A site. Okay, the next process is the process of translocation. What do you mean by translocation? The translocation is a process where the tRNA from the A site is transported into B site, and the tRNA which is present in the B site is transported into A site. This EFG will help in transferring or translocating this structure. Okay, the EFG is also requires GTP. This AFG binds in the A site, so binds in the A site and replaces the tRNA from the A site to the P site, and which is present in the P site, the tRNA will go into the E site. In this way, after the translocation, it is being released in the form of GDP. Now we will see the next process, that is the termination. So for the termination, it requires two factors, which is known as releasing factor 1, RF1 and RF3. When there is stop codon, this RF1 comes and bind in this A site. It is having two sequence, that is GGQ sequence and SPF sequence. The SPF sequence is binding to the mRNA, and this GGQ sequence which is present, this is complementary, or we can tell that it is like the amino acid. After the RF1 has come and bind in this region, RF3 come and bind, which will help. Peptide bond is released from this region. Now, the by using the GTP, this RF1 is also released, and RF3 is after this release of this RF1, this RF3 GTP is also now released. The RF3 is in the GDP form when it is in free form, when it is in free form, when it is being used, it is in the GTP form. In this way, now this protein has been released from the 70S ribosome. In this way, the process of translation in case of prokaryotes takes place. In the next video, we will discuss about the translation of protein in case of eukaryotes. If you like this video, please do like, share and subscribe. Thank you.